Good day, my esteemed students. Once again, we are into the class of Biochemistry 408 or BCH 408, that is, Blood Biochemistry and Immunochemistry. Once again, welcome to lecture number 4, and the topic for today is antigenic determinants of blood groups. Antigenic determinants of blood groups. Let's look at the introduction. We have earlier said that blood is a liquid tissue that is red in pigments due to the presence of hemoglobin molecule, and that all blood are not the same, having specific identification tags on their surfaces. These tags are referred to as blood group antigens. Now, what are these antigens? They are simply proteins or carbohydrates found on the surface of red blood cells, and they vary from persons to persons. They are genetically determined and are responsible for the different blood groups. Let's take a look at the learning objective for this lecture number 4. In the first instance, we will define the term antigenic determinants. Another word for it is epitope in the context of blood groups. Describe the chemical nature of the major blood group antigens. Explain the genetic basis for the expression of ABO blood group antigens. Discuss the role of glucosyltransferases in determining ABO blood group types or groups. We will also go ahead to describe the structure and importance of the resource D antigens. The short form of resource is H, arrow H. Differentiate between the various forms of resource antigens. Understand the concept of immunogenicity in relation to blood group antigens, and explain how antigenic determinants of blood groups are crucial in transfusion and hemolytic disease of the newborn. Now, the learning objective may seem lengthy, but they are very easy to grasp. So here we go. Starting with definitions, let's look at the meaning of the following. Antigen is a term used to refer to any substance that can trigger an immune response. Antibody is a word used to mean a blood protein produced to counteract the specific antigen. Epitope, a small site on an antigen molecule to which a complementary antibody specifically binds. Now, you can liken this to enzyme substrate reactivity. Immunogenicity refers to how a non-self antigen elicits immune response. Phenotype, the actual blood type, which could be A, B, AB, or O. Let's take a look at the current picture shown before us. We can see that there is a cell with an antigen on its surface. Then there is an antibody that is binding to the antigen on the surface of the cell. Now, this antigen contains or may contain small sequences of amino acid or sugar. Now, it is the nature of the amino acid or sugar present on the antigens that determines the type of antibody that will bind to the antigen. So, a particular antigen must express specific amino acid or sugar molecules on its surface, which a corresponding or an antibody that recognizes that amino acid or sugar will bind. So you can have thousands of antigens, but a particular antibody that has a complementary amino acid or sugar will only bind to that antigen. So that binding surface or the amino acid sequence that determines the type of antibody that will bind to the antigen on the surface of the antigen is known as epitope, and that is the antigenic determinant. So antigenic determinants are amino acid sequence or sugars present on the antigen in question. Let's look at the major blood groups antigen. There are mainly two. There are others, but there are mainly two. The ones of concern are the ABO blood group system antigen and the resource factor or resource system blood group antigens. We can now look at the ABO blood group system antigens. The ABO grouping system is classified into four based on the presence of antigens on the surface of the red blood surfaces, red blood cell surfaces, and plasma antibodies. Such that we have blood group A. Blood group A contains antigen A and antibody B. Blood group B contains antigen B and antibody A. Blood group AB contains both A and B antigens and no antibodies A or B. Blood group O contains neither A nor B antigens and has antibodies A and B. Those are the four major blood groups. Looking at the role of glycosyltransferases in ABO blood types. Now, how is ABO blood group is determined? That can be done using the enzyme glycosyltransferases. There are two types, that is the alpha transferases and the beta transferase. Now, for blood type A, the alpha transferase is used to determine the antigen A. Under the situation, the N-acetyl galactosamine is the sugar added. For blood group B, we need the beta transferase, and the sugar added is galactose. The antigen that responds to that is the antigen B. For the AB, both the alpha and the beta transferases are used. And both sugars are added. That is acetyl galactose, N-acetyl galactosamine and galactose. For group O, none is active and no sugar is added. The antigen on the red blood cell surface is the H antigen. Next is rhesus blood system antigen. The rhesus antigen is the third type that may be present or absent on blood cell surface. There are 5 main rhesus antigens on red cell surfaces. We have them as the capital C, small letter c, capital D, capital E, and small letter e. But of all these, the most important is the resource factor D. Now, it is the presence of or absence of this resource factor D that determines whether a blood is referred to as positive or negative. Some of you have the knowledge that you are carrying A plus blood type or B negative. You will understand very soon what that means. In the context of blood grouping, the A, B, and resource factor D antigen and other blood groups antigen have specific carbohydrate or protein structures on the blood cell surface. From the two main systems of blood, four main groups can arise. That is A, B, AB, and O. Classification of either positive or negative blood is based on the presence or absence of the resource factor D. So, like I told you earlier, you may be A positive, A negative, B positive, B negative, and so on and so forth, even O positive or O negative. Now, blood matching. When someone is transfused with blood, the immune system checks if the antigens on the donor's blood is a match to that of the recipient. If it does not recognize it as the same, it treats the blood as foreign and attacks it, leading to severe life-threatening reaction. And of course, this is dangerous. So, for transfusion purposes, blood must match to avoid these consequences. Still on blood matching. In the plasma are proteins that target foreign antigens. Someone with type A blood has anti-B antibodies and they cannot safely receive type B or AB blood. For the universal donor, O negative blood can be given to anyone because it lacks A, B, or resource factor antigen, making it the universal donor type. AB positive individuals. Please take note, AB positive individuals can receive any blood type because their blood contains all the major antigens and no anti-A or anti-B antibodies. Donors and recipients. It is time now to look at the chemical nature of the major blood group antigens. I have mentioned it before now that there are two major types of blood groups, the ABO system and the resource antigen system. The first, that is the AOB, the ABO system, is carbohydrate based. That is, the antigens are mainly carbohydrates, while the resource antigens are mainly protein. As mentioned earlier, the ABO blood group systems are distinguished by specific carbohydrates present on the surfaces of the red blood cells. These sugars are oligosaccharides and they bind to either protein or lipids. Of course, when they bind to proteins, they becomes glycoproteins. And if lipids, they become glycolipids. This slide before you re-emphasizes what was earlier said about the ABO blood system, their compatibility as well as their importance. It clearly shows who can receive blood from whom and the type of antibodies or antigens present. Now let's look at the ROH blood group antigen. I told you that the resource factor system, particularly the D antigen, is crucial for blood type compatibility. So when we talk of resource factor or resource antigen, the antigen D is most crucial. The slide here presented shows the type of antigens, that is the capital C, small letter C, capital E, and small letter E. Then the genes that are responsible for this type of antigens, the ROHD, resource factor D, resource factor C and E for either capital C or small letter C. And you have the same as for capital E and small letter E. Now, the presence or absence of resource factor D gene and the amino acid that sequences associated with them are presented. Then this table also shows the importance of these resource factors in transfusion. Again, this is this slide on the resource factors antigen D, capital C, small letter C, capital E, and small letter E, emphasizing what was earlier said on these antigens and their risk factors. In the previous or even in this slide, you can see HDFN, which is hemolytic disease of the fetus and newborn. Hemolytic disease of the fetus and newborn. So this is a summary of the 5 main resource factor antigens, their genetic basis, significance, and antibody risk. Looking at the hemolytic disease of the newborn, now this occurs when maternal antibodies attack fetal red blood cells due to ABO blood system incompatibility and the ROH blood group incompatibility. That is, when a pregnant woman is carrying a baby and there is incompatibility between the baby's blood cells and the mother's blood cells in terms of ABO system and the resource group system. So complications can arise from here. Now, when a mother with blood type O is carrying a type A or B fetus, the maternal immunoglobulin G anti A or B antibodies cross the placenta but cause a milder hemolysis. This is because the fetal red blood cells expresses fewer ABO antigens. The antigens are also present in the fetal tissues, thereby diverting antibodies. In most cases, this scenario requires no treatment. However, severe instances may arise that will require phototherapy or exchange transfusion. Resource negative mother carrying a resource positive fetus may develop anti-D immunoglobulin antibodies after fetal maternal hemorrhage. These antibodies cross the placenta in subsequent pregnancies, causing fetal blood cell hemolysis or lysis that will result to anemia. Now, having said all that needs to be said about lecture 4, you can use this opportunity of these questions to test your understanding of what has been presented. It will do you good to go through the slides over and over. I mean, it will do you good to go through the slides over and over so that you can completely comprehend what is presented. I have 5 questions here to test your understanding, and I hope that it will serve the purpose. Thank you and have a nice day.